everyone, and welcome to today's episode on respiration exam questions. You know the drill by now. I'm going to show you from start to finish how I'm thinking, approaching, and answering the questions. So let's begin. The one I picked up today is very interesting. I would recommend you having seen or spoken about this practical in class before you watch this video, because then it'll be a bit of a waste, but let's get into it. So, how I'm going to think about it is that I know that I'm not going to read anything except the exam question first, because then it stops me from overthinking the answer. So let's do that. Give one limitation of using this method to investigate respiration rate. Okay, so I know it's going to be a limitation, and it says it's about the method. So what I'm going to do is I'm going to go to the method, read it, and see the potential problems with it. However, I also do know that there are stuff that are always on the mark scheme for this type of question. And without reading anything, I'm immediately thinking of number one, like the seal not being airtight, and number two, the time measurement. I would probably say make it more frequent. And I know to say about time because it says respiration rate. You tell me, what does rate mean? It means 1/time. So we're involving time into it. And as scientists, as biologists, we like very close and frequent measurements to be taken. So if it was every 1 minute, you'd be like, make it every 30 seconds. If it's every 30 seconds, you say make it every 15 seconds or something like that. That's the thought process. And I thought of all of those points, A, from doing exam questions and knowing that these things actually always come up, but B, without even looking at anything, all I know just from a glance of the question was that it was going to be on this practical, and that was it. I never read anything else. But now it's time to read. So this is the method here. Now if we read this, we can see that soda lime is in a syringe, there are germinating mung beans. We can also see the movement of the red fluid is being measured, and then it gets reset. Okay, well, I think my answers that I already said are enough, to be honest, because when we say, oh, the seal might not be airtight, we say that so that gas doesn't escape, and we don't want gas to escape, because then, it would just ruin the experiment, because the movement of fluid is affected by it. Another thing, looking at time right now, is every 30 seconds. What did I say? The scientist, we want closer time intervals. So it says only one limitation. I've spoken a lot about it, so let me just put one of them. Okay, next one says, identify one variable that has not been controlled and suggest an improvement. Oh, I love these questions. It's the same every time. You will understand there will be a pattern in practical questions, because let's be honest, nothing really can change except the actual practical that's being done. So I know that is talking about a variable that's not been controlled, so we're looking for a control variable. How can we identify it? Well, we know if we look to the method, if something or measurement is not given, then that is, it hasn't been controlled, So let's look here. Immediately before reading the words and just by scanning, I can see there are no mention of numbers or centimeters cubed and stuff like that. So I already know there are going to be a lot of variables that have not been controlled. Let's start with the first bullet point. It says a bag of soda lime. How much? If you did this experiment, would you know how much soda lime to put into the bag? I wouldn't, so you wouldn't, no one would, right? You need to specify this. So that's the variable that has not been controlled. The improvement for it would be very simply, use a known mass of soda lime each time. Very simple. Let's have another go. It says 5 germinating mung beans. Do you know what mass the mung beans should be? Do you know whether they should be the same size or same height, stuff like that? You don't. So that's why the variable that's not being controlled is the mass of the seeds. It's just simply not given. What can we do to improve it? Well, just take the mass of the seedlings at the start. Okay, next question says, describe how you would add the red fluid in the capillary tube at the start of the experiment. This is what they're talking about here. I'll be completely honest with you guys. Just learn it. Don't think too hard about, how do they do it? Because when I first attempted this question, to be honest, I literally did not know what to put. Like, I knew and I saw how it was done, but to put that into words was a bit hard. And so you basically just memorize and put in your notes and say, well, it's dipped into a small beaker and allowed to run. Next question is all about anomalous results. This is a very, very key concept to understand in biology because when anomalous results come up, something's gone wrong. And if something's gone wrong, you need to really do something about it. But we'll get onto that. It says, explain why the result is considered to be anomalous and describe one correct way of dealing with this type of result. An anomalous result is when it just doesn't fit with the pattern or it's just out of line, really. 17.5 doesn't really line up with the values going across, and it also doesn't line up with the values going down. And then the action you would take would just be repeat at 60 seconds to obtain another reading, because you want to see whether it was actually true or was it like a mistake, and then you now get the new results from doing the repetition. Repeats are so important, remember that. What additional information would be needed to calculate the volume of oxygen taken up by the seeds? So we're talking about volume here, and we have a capillary tubing, okay, which we consider basically as a cylinder, really. Now, you're given the length, you're not given the radius or the diameter of the capillary tubing. So how are you going to calculate the volume? Volume of a cylinder is pi r squared times by length. We have this, well, pi we're just given. We don't have this. We need the diameter or the radius, whichever one you like to say, of the capillary tube. Next one says oxygen uptake for this batch of seeds to be comparable with data from another type of bean. So, interesting, isn't it? So if we want something to be comparable with data of another bean, what do you really need as an information to calculate? Well, you're going to need the mass of the bean seeds, because that's the only way it can be comparable. Yeah, the type's different, but something needs to stay the same so we can keep the mass the same. Okay, and that's the end of today's episode. Remember, the two most important exam techniques are read the question first and state all the obvious points. Now, over to you. Try some questions and let me know how it goes.